Thank you very much, Quentin, for agreeing to give a Build a Cell seminar, and you can get started.
Thanks so much, Kate. Really excited to be here. I've been following the Build Cell Seminar for a long time. Absolutely thrilled to be talking about my experience and what we're going to be doing in the Dudley Lab going forward. So to introduce myself, I've been interested for a long time in this idea of metabolic engineering to harness biology to generate sustainable chemicals. And those could be low value things like biofuels or commodity chemicals and polymers, or high value things like medicines and pharmaceuticals. And so this interest really started for me during my PhD at Northwestern University, where I was in Michael Jewett's group and did a lot of prototyping using cell-free protein synthesis. You'll see that later in the talk. But I wanted to broaden outside the bacterial mindset, and that's where I did my postdoc actually in the UK. And that's where I first got into plants and the amazing chemists and the secondary metabolites that they're able to make. But following my postdoc, I actually spent time in industry as well as in science funding, thinking about how to apply these ideas practically. And then it was in early 2024 that I joined the faculty here in the Chemical and Biological Engineering Department at the University of Wisconsin. So to zoom out, I'm really excited about plant synthetic biology as this new frontier for biomanufacturing with the potential to accelerate a transition to a distributed, sustainable, and post-fossil carbon society. And that's really just about the ability of agriculture to scale things with potentially lower capital costs than the bioreactor-based production mindset that people typically use for microbes. So what are some of the exciting areas that I see in this space? One is just making plants have higher flux, more biomass. There's some really cool ways to think about how you can engineer metabolism to increase soil carbon sequestration. For example, there's about 450 gigatons of carbon in plant biomass, which is 200 times more than all the animals on the earth. There's been long-standing goals of engineering plants to fix their own nitrogen or remediate contaminated soils. But then in the more synthetic biology mindset, maybe we're thinking about sentinel plants that detect environmental cues or even respond to them by creating agrochemicals in situ. Or the theme of this talk would be how we're using plants to produce novel medicines. So my talk outline today is going to be in two parts. One, I'm going to tell you the story of my postdoc, where we were using genome engineering in this wild tobacco called Nicotiana benthamiana to produce a class of molecules called monoterpene indole alkaloids. And then at the end, I'll shift to what the Dudley Lab is going to be doing at UW. So to start, again, my postdoc was a joint project between Nicola Patron, she was at the Earlham Institute, and Sarah O'Connor at the John Innes Center. Both those were co-localized in Norwich here. And since then, Sarah's moved on to the Max Planck for Institute for Chemical Ecology in Germany, and Nicola recently accepted a position at University of Cambridge. And so the goal of my postdoc project was really looking at this molecule, vinblastine. It's a potent chemotherapy drug. It's a microtubule inhibitor, frontline treatment for Hodgkin's lymphoma and other cancers, and it's naturally made in this plant, Catharanthus roseus, commonly known as the Madagascar periwinkle, which produces this as well as 130 other similar monoterpene indole alkaloids, but it does so at extremely low levels in the plant, making sourcing from this natural environment costly and difficult. So this metabolic pathway to vinblastine is also extraordinarily complicated. It's 29 dedicated steps, with the last ones here being elucidated by the O'Connor group back in 2018. And so my goal, looking at this complex metabolic pathway, was to look at the first half and see what we could do about getting to this intermediate molecule, strictosine. And that's because strictosine is the branch point for the over 3,000 monoterpene indole alkaloids. And I mentioned vinblastine as a cancer drug, but there's a number of other relevant or therapeutically relevant molecules that a metabolic overproduction platform for strictosine would potentially open up. So Prior efforts and maybe the conventional mindset on doing this would be to take the individual genes encoding this metabolic pathway and express them in a microbe, for example, a baker's yeast. And this had been done by a postdoc before me in the O'Connor group, but they really did struggle to get high yields. And we hypothesized that this is due to the difficulty of folding these enzymes that evolved in a plant context. So rather than using a yeast, my goal was to reconstitute this pathway in a plant host. And there's some preliminary work done by a consortium group in Belgium, Netherlands, and France that had been able to do that, but had to split the pathway up into two parts. So the plant that we were going to be using is called Nicotiana benthamiana. Again, it's a wild tobacco relative native to Australia, and it's a really useful bioproduction platform because of a mutation in its immune system that makes it susceptible to agrobacterium-mediated transformation. And so the idea there is you can take your gene of interest, put that on a plasmid, and then use this shuttle bacteria to syringe liquid into the interstitial leaf space, and then the plant will transiently express your protein 5 days later. And some fraction of it's incorporated into the genome, but in this way, you can activate multi-step metabolic pathways rather than having to put all your genes on a single plasmid. What you can do is just put each pathway enzyme in a separate strain of agrobacterium and co-deliver those and deliver up to 15, even 25 enzymes simultaneously. And that means benthamiana has been a really useful plant for making medicines, but also elucidating biosynthetic pathways. And I'm putting a small subset of the plant metabolites that have been elucidated using this type of approach. So yes, we can figure out plant pathways, but how can we make them? Well, benthamiana could also be a scalable bioproduction platform. So rather than using a syringe to push those agrobacterium, you can take the whole plant, dunk it upside down into this bath of agrobacterium and vacuum infiltrate to transform all the foliar leaf tissue. And then think of more like an industrial greenhouse to produce pharmaceutical proteins or metabolites at scale. And the number of companies have tried to do this with some exciting efforts in making both therapeutic antibodies and rapid response vaccines, including a COVID vaccine, which was produced and approved for use in Canada. So I wanted to produce streptozin and benthamiana, but the challenge was that yes, benthamiana is a great host for expressing proteins and providing the precursor GPP and tryptophan that we'll need. But on the downside is there's all this other metabolism happening in the plant. And that was interfering with our plant, our heterologous pathway. And so what we would see as we started to express these early enzymes was modification of these intermediates. For example, with the addition of glutathione, overoxidation, a lot of glycosylation, and then acylation on these compounds. So how I could see that would be on a liquid chromatography mass spec trace, where here's the empty vector, but then you add in these early enzymes and all of these unwanted peaks are appearing. So that led to a three-part strategy for enabling streptozin production in this plant. First, I worked on knocking out some of these enzymes that are causing these unwanted derivatization via CRISPR-Cas genome engineering. I also will briefly talk about our efforts to remove a co-purifying contaminant metabolite, and third, to increase flux through this pathway by adding a missing step. So to get started on the genome engineering side of things, the first challenge was to figure out what enzymes might be actually doing these transformations. And the first challenge was that when I started this project in 2017, the draft genome of benthamiana was highly fragmented and difficult to figure out exactly how many candidate genes were there. Because it was built only from short read sequencing. So our first effort was to work with DNA assembly experts and incorporate some long read sequencing to build our own improved genome. And around the time that we published our work in 2023, there were a number of other efforts that have also come out, which are quite good and maybe probably better annotated than the genome that I used. So great, we actually knew how many candidate genes we were working with, but how can we identify them? So we did some transcriptomics experiments to see which genes might be upregulated when the metabolites are accumulated. We also did a lot of phylogenetic comparison, trying to look at these starred and diamond shapes. These are glycosyltransferases with known activity on similar substrates, so maybe closely related benthamine glycosyltransferases could be good candidates. But we also wanted to do some in vitro activity screening. And for that, we collaborated with the Ehrlich Institute BioFoundry to do some liquid handling automation and build on the techniques of cell-free protein synthesis that this audience really needs no introduction to. So, as you well know, the approach for crude lysate cell-free protein synthesis is to break that cell open and then add amino acid building blocks for your protein, NTP building blocks for your RNA, and an ATP regeneration system, as well as a DNA template encoding your protein of interest. And so what I added to the community was there was a whole library of standardized Golden Gate DNA parts that the plant community has adopted. And as we were working with our pathway, I wanted to make it so that I could use the same building block parts in both a plant-based expression system and a cell-free expression system. So I built a toolbox of DNA parts and acceptors, N-terminal expression tags, C-terminal detection tags, and several reporter proteins to make a resource for my own efforts as well as for the community. And the overlaps between these DNA parts match the standard that the plant community is using. So with that, I really started to focus or also used the Ehrlich Institute's BioFoundry with a lab site Echo, which can do 384 to 384 transfers of liquid. And I was using this both to assemble my DNA and also to assemble my cell-free reactions. And we focused on the glycosyltransferases and showing that we could assemble a number of these benthamine UGTs, but some expressed well and required some N-terminal solubility tags. Some, really worked better with, this tag, but some just didn't express well at all. But the exciting thing was that those that did express, we could then feed our substrate molecules directly and measure on a mass spec if there was activity on that particular compound. Then we would know if this was a good target for knockout. So the combination of these four approaches then left us with 34 candidate enzymes. And then we were off to try and knock them out. So if you're not familiar with plant genome engineering, it can be quite laborious. So what I'm showing here is a vector that we would deliver to a plant using that agrobacterium shuttle. We have a selectable marker, the Cas9 nuclease, as well as the guide RNAs telling that nuclease where to cut. And the typical approach then, if you want a stably transformed plant, where it will have this construct and it'll be heritable to multiple generations, would be to deliver this to an embryonic callus, wait some amount of time on selection, then add a set of plant hormones to do rooting, then a different set of plant hormones to cause shooting. And it's a lot of work. And without the efforts of my collaborators, we would not have been able to do as much of this as we did. However, halfway or in the later stages of my postdoc, a new technique came out actually from the University of Minnesota and Dan Voitas's group, where they were able to do knockouts in benthamine that was already expressing the Cas9 gene. So the downside is that you already have to be expressing Cas9, but the upside is you can deliver your guide RNA that has this mobile epitope on it that then, by delivering with this viral sequence, you can transiently express the virus. And then the RNA will move through the plant vasculature to the meristem, make an edit, and then all the leaf tissue above that point should be edited. And this was so much easier. And I made as many knockouts in 6 months with this technique as in 3 years with this one. And so the combination of all this effort was that for those 34 candidate genes, we ended up with 18 lines with stable mutations after genotyping 300 T0 plants. And so the table showing this here for the GTs is here's the different line names, and this lists the knockouts we made. A single knockout here, a couple quadruples, and so on. So this let us then test the effect of these knockout mutations. So, as I mentioned earlier, we were caring about these derivative peaks over here as we infiltrated into the plant each of these pathway enzymes. So these are the things that we're trying to get rid of. And we tracked 20 peaks, looked at 11 knockout lines in this table, and then quantified peak area. And so what we found is that a lot of the peaks remained. And that's what we are hypothesizing, is that these are notoriously promiscuous enzymes. So maybe knocking out just one or even a handful wasn't sufficient to remove all the peaks. But we did have a few isolated successes, showing that this group A UGT meant that these peaks disappeared for peaks 3 and 5, and this A85, A73 successfully removed peaks 1, 2, 4, and 5. So we validated the hypothesis, but weren't quite as successful as we had hoped. And ultimately, the knockout lines we made didn't help our overall product accumulation as much. Something else was ultimately limiting. So, but while we were doing all this knockout work in Benthamiana, we also thought of the idea of, well, the plant is also making a lot of this metabolite you're probably quite familiar with, nicotine. And that's going to be a problem for bioproduction, as it's going to co-elute and co-purify if we were to be successful in reconstituting monoterpene indole alkaloid biosynthesis. So while we were knocking out these GTs, we also wanted to knock out nicotine biosynthesis. And to do this, we focused on the final hypothesized enzyme, this berberine bridge-like enzyme, which was, again, hypothesized to do the linkage between the 6-ring nicotinic acid and the 5-carbon N-methylpyrrolinium ion to make nicotine, and also is probably making these other alkaloids here. So in collaboration with Fernando Guy Flores's group at the University of Copenhagen, we targeted BBL for knockout. We initially went after 3 homologs that we could find in the plant, but we're still making nicotine. So we realized with our improved genome that there was actually 4 homologs of the gene plus a pseudogene. So we finally got our quintuple knockout, while we were able to get anatapine to go to 0 as well as anabasinine, we really were still making nicotine in the end. So here was our wild type lines, here's our triple knockouts and the quintuple. And it wasn't until Fernando's group had the idea of actually running this assay on a chiral column that we found something really interesting, that the nicotine made in the wild type plants was all S-nicotine. But the nicotine made in the knockout lines was racemic, both S and R nicotine. And so even though we didn't succeed in making a nicotine-free plant, we've actually got some nice insight on how nicotine biosynthesis actually works. And Fernando's group developed 2 competing hypotheses of how BBL is actually playing a role in determining the stereochemistry of this biosynthetic reaction, maybe just as being the coupling enzyme itself, or determining the stereochemistry, but pointing to another potential missing enzyme. So, but it was approach 3 that ultimately led to success in this project. And that was to increase flux by completing the pathway. And so ongoing work in the O'Connor lab at the time was looking at a related plant species that also produced alkaloid molecules, but not strictosine. And this set of plants was the Nepeta genus, so commonly known as catmint or catnip. So this is Sarah O'Connor's cat. We tried to get it on the journal of the cover, and ultimately that they did not do that. But the idea behind catnip is it makes these Nepeta lactone iridoids, depicted here. And there's actually overlap with the biosynthetic pathway for these early steps highlighted in yellow. So Benji Lichman, who's recently started his own group at University of York, was working on this and had discovered a new enzyme, this major latex protein-like enzyme that was really essential in establishing the stereochemistry of the nipidolactone in molecule in nipida, as well as accelerating the rate of this step. So my question was, well, could MLPL help us in this metabolic engineering project? And it turned out it was really helpful. So what I'm showing here is, again, those mass spec traces where we're putting just the first 3 steps of the enzyme, of the pathway, the first 8 without the MLPL. And then as you add the MLPL, you see this new peak accumulating, which matches the 7-deoxyloganic acid standard that we were intending to. And so this was ultimately the secret sauce that adding in this MLPL let us reconstitute the entire pathway in the plant. So what I'm showing here is we had that early pathway all the way to 7-DLA, and then we could add each subsequent enzyme one at a time and see the accumulation of 7-DLA. But then as we add another enzyme, that peak goes to 0 and we see loganic acid accumulating, then loganin, and finally strictosine when we added the entire pathway. And ultimately, we were able to make 200 milligrams per kilogram dry weight. So not a ton, but a great proof of concept for future metabolic engineering. So in summary of this story 1, I've talked about our 3-pronged approach for improving strictosine production in bentamiana. 1, to use CRISPR-Cas genome engineering to knock out derivatives, enzymes causing unwanted derivatization. An effort to create low nicotine lines for bioproduction, and then the key of this MLPL enzyme that just came out. And I'll highlight actually just 1 month ago, the O'Connor group published a paper showing that yes, Catharanthus roseus also has an enzyme mediating this interaction, the so-called iridoid cyclase. But it took some really clever work by Maita and colleagues to figure out what that enzyme was in the native plant. So I'll then wrap up this part 1 by acknowledging both Nicola and Sarah and a number of collaborators and colleagues in those 2 groups. So with that, I'll shift to what the Dudley Lab at UW is going to be doing, and we're going to be focused on developing tools for cell-free and plant synthetic biology, really going back to this vision of plants as bioreactors that I motivated this talk with. So being here for just my 4th semester now, we've got a great cohort of graduate students that is anchoring the group, and a number of undergrads that have made some nice contributions. So the Dudley Lab is going to be working on 2 different project areas. One is going to be more plant-based, looking at that one of the things I noted during my postdoc, as well as experience in industry, was the lack of tools for controlling enzyme expression, especially if you want to not transiently express your enzyme pathway as we were doing in Nicotiana bentamiana, but if you want to stably express your metabolic pathway in an agronomically relevant crop. And so some areas in the lab that we're going to be working on is how can we redirect metabolic flux using some of those higher throughput genome engineering techniques I mentioned, as well as genetic toggle switches, perhaps mediated by either inducible promoters, or in this case, we're looking at serine integrases that can swap the orientation of a promoter element to have in this case, this FPPS gene will be on, and in this case, this will be off. And that'll help us control more terpene flux going to geraniol and the molecules we want, rather than supporting cell growth and development. So this is something you would want to turn on only when you're ready to move from a cell growth phase into a bioproduction phase. Another project area here that I'll talk about briefly is how we're understanding the transfer DNA architecture of what is encoded in that region of DNA that the agrobacterium is delivering to the plant. And what we've noticed is that existing DNA sequences for regulating gene expression, such as promoters, are inconsistent and context dependent. So, for example, here's a really nice strong promoter that works well in isolation. You get lots of enzyme B, but as soon as you put it on a multiple transcriptional unit vector, there's these unknown interactions, either through repressors or enhancers, that now cause this to be a very weak promoter. And so I tried at length to put as much of the strictosine pathway onto these multi-transcriptional unit vectors and ultimately never got all enzymes to express simultaneously. So the way that we're going to address this, and we've got some preliminary funding from both Wisconsin and Bayer to look at this, is a high throughput readout where we're co-delivering pools of different architectures of this transgene, where we can change things like the promoters and terminators used, the presence or absence of insulator sequences here, as well as the order of genes and the orientation of genes. And we're imagining using liquid handling automation to make hundreds of, or even hundreds of thousands of gene variants, pooling those together and delivering them to the plant. But then each gene will have an RNA barcode that we can link back to the original vector with long-read sequencing, and we can look at the relative profiles of these barcodes to say, wait, this was maybe a good vector. None of the enzymes was going to be rate limiting, where we might not want to use something bad like this. And initially, we're going to be screening just for a vector that works, but I'm really excited for this approach to really elucidate the molecular mechanisms underpinning these interactions, potentially with some collaborations on machine learning and modeling. So that's the plant side of the lab that we're spinning up. We're also leaning into the fact that there's still a limited knowledge of plant secondary metabolism and regulation. And how can our cell-free systems help increase the design, build, test, learn cycle for plants? So hopefully what you've already seen in this talk was how cell-free protein synthesis was able to guide genome engineering. I also worked during my postdoc to help understand native gene regulatory networks by using cell-free protein synthesis to express transcription factors so that we can do in vitro binding assays to understand which regions of DNA those transcription factors are binding and validate hypothetical gene regulatory networks. I'm also very excited to use cell-free protein synthesis for enzyme discovery. As I maybe alluded to a couple times in this talk, Benthamine is a great platform, but you're throughput limited, and most postdocs or researchers in this space only have the bandwidth to test, maybe 50, at most 100 candidate genes. Well, how can we use cell-free protein synthesis to screen not hundreds of genes, but maybe even thousands, coupled with low-cost gene synthesis? Also, I'd like to explore ideas for how can we optimize pathways and find which enzymes are going to, or enzyme variants are going to be most useful or most active in isolation or in combination before going through that laborious tissue culture to deliver these genes to a stable plant, or to even do enzyme engineering to improve the activity of these plant enzymes, which didn't necessarily have a ton of selective pressure for high activity in their history. So for that, we need cell-free systems that can express plant proteins. And so we're using both what I'm calling kind of a bottom-up approach, using the well-established E. coli-based systems and trying to make them more plant-like. We're also using the plant-based cell-free systems, either derived from wheat germ or a new system derived from tobacco BY2 cells, as more of a top-down approach. These already should be good at expressing plant proteins, but have different complexities experimentally than using the E. coli system. And so one of our first efforts has been to validate and compare or benchmark these different systems for not just reporter proteins like GFP and luciferase, but actual plant enzymes. And so that's what one of the first students in my group, Sarah, has been doing. So she's established the E. coli cell-free protein synthesis workflow here in Wisconsin. We're making some GFP, and we've been testing both the commercial wheat germ kit as well as the tobacco BY2 cell system. Last summer, I had a visiting scholar with experience cultivating this is one of the most common plant cell lines used in plant research. And so we've been working on both using the commercial iteration of this and looking at reverse engineering our own version to potentially further improve it for making plant proteins. And the long-term vision here is to do a lot of what I did in my PhD in the Jewett lab. And I know Ashti Karim and I co-developed this. He gave a build a cell just a few weeks ago. And we called this optimization approach iProbe, where you could express different pathway enzymes in different cell-free protein synthesis reactions, and then mix and match those together to optimize a metabolic pathway, and then that insight could help you then engineer whichever organism you were actually engineering. So an iteration of this talk could have been how we make limonene, another plant-based molecule of fragrance and flavoring found in lemons as well as several other organisms. So Sarah would like to do this type of mix and match approach for something like the monoterpene indole alkaloid pathway. But we're challenged by the fact that even just that first 14-step pathway that I was working with contains 4 cytochrome P450 enzymes. And those have been a notoriously difficult class of enzymes to heterologously express. And so Sarah's working on a couple of hypotheses of how we can make the E. coli cell-free protein synthesis system better at expressing P450s and also the cognate reductases that are needed to actually take NADPH and then take those electrons so that we can have our P450 be active. And so some of the things Sarah's exploring is supplementing our reactions with small amounts of heme and controlling its degradation, adding membrane mimics, as well as some chaperones. And so in future, we're looking at maybe how can we use this iProbe type approach to find which P450s are best matched with their cognate reductases. Another exciting direction I'm hoping to take the lab is rather than to build expensive things like strictosine, how about we build low-cost things like these polymer precursors? P-hydroxybenzoate and 4-vinyl phenol. So these are aromatic polymer precursors, and the polymers they make are somewhat higher value than polystyrene. But this is a collaborative project that we've nominally been funded by through ARPA-E, with the lead PI being Hiroshi Maeda here at University of Wisconsin in the botany department. And we're also collaborating with Victor Zavala in the CBE department with me, as well as the Wisconsin Crop Innovation Center. And the idea here is that we're going to be metabolic engineering both the production of precursor amino acids as well as the downstream enzymes that actually make these molecules. And we're going to be doing these both in a more of a test bed system, the Nicotiana tabacum. This is the tobacco that you're probably more familiar with. That people smoke, but the long-term vision is to put these metabolic pathways into a bioenergy crop like sorghum. And in that way, you can have a value-added stream that you can chemically extract these molecules before taking the remaining sorghum biomass into a cellulosic ethanol pipeline. So at the risk of putting too complex of a metabolic pathway up here, Hiroshi's group is an expert in aromatic amino acid biosynthesis in these enzymes in blue. So we have a number of versions of those that are feedback insensitive. And so we're hoping to be able to make lots of amino acids and using cell-free systems to screen which metabolic enzymes are going to make our product. But what we also want to do is engineer or screen glycosyltransferases that will glycosylate these molecules on purpose so that they will be sequestered in the vacuole and avoid impacting plant growth and development. We also have a component of this project that would be working on some of those metabolic switches that I mentioned earlier. So that's where I'm going to wrap up my talk today. I just also want to shout out my department and the really exciting things that are happening here in terms of new facilities and new research group, which we've hired 9 faculty in the last 5 years, including me. And I'll take any questions that we have time for.
Thank you. Thank you so much. There are questions in chat. Can you see chat?
Let's see. No, but maybe
I could read you questions too, if that helps.
Sure. Let's do that.
Okay, so question from Andre. Could you please elaborate what's the unusual challenge of plant-based system DNA parts versus other cell-free systems?
I hope potentially that was when I was talking about the library of vectors here.
Yeah,
So I'm
that
going
was
to
looking at the
interpret that.
timing that,
Yeah.
yeah.
So there's really this was an effort of convenience that I'm hoping to, as we both order genes for our own lab and utilize and collaborate with folks that have libraries of genes already in this standardized Golden Gate format. We're hoping that then we can use those coding region parts directly in a cell-free system. And so what are some of the challenges? It kind of relates to what I was alluding to with the challenge of expressing cytochrome P450s. Just in that not all plant enzymes are well behaved and soluble if you just put them in with a start codon right here and a stop codon in the end. And that's where we found that a number of solubility tags were helpful for some plants, but not for others. And I think that points to either, high throughput approaches to start to understand what I would call the folding problem, or needing to do these high throughput screening approaches to just find a solubility tag that works and move forward with that.
All right. If anyone has follow-up questions, you can ask here or unmute yourself. Okay, next question. This might be a naive question. Is biomanufacturing in plants better than regular bacterial bioproduction, mostly because plants make their own carbon via photosynthesis, or are there other plant advantages over bacterial?
Yeah, I think that's a very nuanced question, and I'm here to both champion plants, and so I'll maybe start with what I think are the advantages. And I think it is scale. I think we already have agricultural systems that are processing billions of tons of biomass. And so that's a lot less steel in the ground. Not that agriculture is cheap and land is, always at a premium, but I'm very excited for applications that leverage that scalability. Maybe I'll pull up my road mapping exciting slide here to kind of cue on that. I'll also say that when it comes to making vinblastin, there was some really tour de force work by Michael Jensen's group and Jay Kiesling's group to actually get that pathway to be reconstituted in a yeast. I think I have the citations on a slide here. Some really, really great work there. They've also made really trace amounts, and have struggled with expressing all of these plant enzymes in a yeast. And so I think, maybe for shorter pathways or for, it really depends on your goals. There might be some benefits to good manufacturing practice for microbes. But I think the fact that you'll just have a different set of challenges in engineering a plant as compared to a microbe, I think these are both platforms that we'll need in our uh, toolbox moving forward.
Thank you. I'm really happy to hear that as a bacterial TXDL person. Okay, next question from Jesse. Did anyone manage to preserve photosynthesis in cell-free? Add chloroplasts to cell-free extract for energy and carbon fixation.
I believe that has been done. Uh, Particularly just with light and being able to produce NADH. The main references I would point you to the work of Tobias Erb on some of their synthetic carbon fixation platforms, but I'm sure there are many others that he was building off their work as well. So um, I think then, yeah, I that's just exciting spaces. I don't have active plans to do that type of work, but yeah, I think pointing towards, I mean, or building off what Kate just said, we're talking about living organisms right now. What is the future is maybe taking the best of plants and the best of microbes or enzyme reactions in vitro, and I think that's a long-term direction the field will go.
Thank you. Next question from Claudia. What about phytoplankton? Could that combine bacterial advantages with plant benefits?
Sounds great to me. I One of the things that I'm starting to think about is the chat that there's a lot of benefits in plants with compartmentalization. Something I glossed over, but have the pathway in front of me is this pathway, even just this first half, is in 4 subcellular compartments and 2 different cell types in the native plant. Why is that? Could be accidents of metabolism, or of evolution, but more likely there's reasons to keep these different metabolic reactions compartmentalized to avoid crosstalk with either other cellular bioprocesses or with other enzymes in the pathway. And so, I think other organisms, maybe plankton, or maybe, it's these plant cells that we can start to control that subcellular localization. I do think that will be a again, a tool in our toolbox to engineer these pathways.
Thank you. That sounds really exciting. This is the end of the chat questions. Does anyone else have any questions they would like to ask? Going once. And thank you.
Doctor, hi.
Oh, hi, Olivia.
Hello, Kate. Please, could you comment on scaling up? How feasible it would be scaling up protein, heterologous protein?
If I understood your question, I think you wanted me to discuss the scale up aspect of using plants.
Yeah,
Okay.
please.
Yeah, I mean, I think it's not, what do I want to say? The advantage is you don't need to have more steel in the ground. You just need bigger greenhouses. And I think that's where the exciting space of plant-based bioproduction is. And that's where some of the references I had up here for some of the rapid response vaccines. And I showed a picture of some of those greenhouses. I think there, the transient expression by dunking your bacteria or your plant in a bacteria, like that does add handling costs, and then you're still going to have a downstream processing workflow, just like you would need for a microbe. You're going to need to break that cell open, and that might be a little harder than a microbe, but then you're still going to need to isolate your protein of interest. So I think, that transient approach might be feasible for pharmaceutical ingredients where cost of goods is less essential. But I think to build towards lower value and higher volume compounds, what you're going to need is you're going to need your pathway stably expressed in a heritable manner so that all you need to do is take your seeds of the next generation and not do any more gene manipulation at that point. And then it's going to be a question of how are you going to separate your cell growth phase from your bioproduction phase, or at least avoid unwanted interactions there. Maybe that's with inducible promoters. Maybe that's, quote, wiring up these enzymes in gene regulatory networks where they only get expressed, as the plant is nearing senescence. So that's where I've been paying close attention to the gene circuit literature and thinking about some project areas there. But then again, you are now in some downstream processing regimen, and I think it will require careful collaboration with agriculturalists, with chemical engineers, to make sure that the downstream processing is easily able to extract your molecule of interest and still and not have you dead in the water in terms of cost there. And so that's where I was, again, privileged to be a part of the ARPA-E proposal, where we were also working with sorghum producers to validate some of those back of the envelope questions before just diving in and do something cool with biology.
Great, thanks for your comment. Yeah, because the pharma industries, they usually don't like very much soil, isn't it? I mean, it's quite be a very interesting alternative and a very interesting tool for we to produce synthetic or heterologous proteins, and even synthetic proteins, and be accepted by the pharma industries. Yes, thank you so much.
Yeah. No, and again, I think it's each platform is going to have its own challenges. I just saw a paper a few weeks ago looking at, just purifying pharmaceutical proteins, and they were like, oh yeah, a little bit of Rubisco came along in our final prep. And like, oh, is Rubisco going to be any worse than, some E. coli protein or some CHO protein, but it was like, oh yeah, I guess you would kind of expect that, but no, they actually showed it, trace amounts, but we're not used to regulating based on those types of contaminants. So there is a barrier to entry there.
Yeah. I think you pointed out correctly that this is going to be a problem in other novel bioproduction systems too. So that's something we all have to think about.
Yeah.
Yeah, indeed. Yeah. Sounds great.
All right. Thank you so much. If there are no more questions, I want to say thank you so much, Quentin, again. And thanks everyone for a great discussion.
Great.
And
Thanks for the opportunity to be here.
Have wonderful rest of your Monday. Bye.
Bye, Kate. Amazing seminar. Bye.
Thanks.